Welcome back, friends. In today's session, we are going to discuss about Hammaring experiment. Hammaring experiment is sometimes called as Hammaring acetabularia grafting experiment. And this is the experiment which provides the evidence for the location of genetic material in the nucleus. So it's the experiment which was done so as to provide the evidence for the location of genetic material in the nucleus. And this is sometimes called the evidence from green algae. So when we are studying the evidence for the location of genetic material in the nucleus, one among the evidence is evidence from green algae, or sometimes it is called as a Hammaring experiment or Hammaring acetabularia grafting experiment. So the name Hammaring acetabularia grafting experiment, Hammaring is the scientist who performed the experiment. Acetabularia is the genus of microorganisms which used in the experiment, and grafting is because of the procedures of the experiment involved grafting of different parts of acetabularia, as how we shall explain later. So who is Hammaring? First, let's start seeing who is Hammaring. His full name is Dr. Joachim Hammaring. He was a pioneer Danish-German biologist funded by Nad German, who determined that the nucleus of a cell controls the development of an organism. So, since the nucleus of a cell controls the development of an organism, then he said that the genetic material, they are located in the nucleus, because what controls the development is the genetic material, or they are the genes. So some of the books they write that he was a German biologist, but as you can see here, he was a pioneering Danish-German biologist funded by Nazi Germany. That's why books they are writing at the German biologist. Now, one among the things which he founded is that the nucleus is the location of genetic material. Now, how he demonstrated that nucleus is it controls the development of an algae or the development of an organism. Now, in 1934, he demonstrated that nucleus determines the character of a cell, and ultimately the characters of individual. So if the nucleus determines or controls the character of a cell and the character of an individual, that means nucleus contain hereditary material. In other words, cell store hereditary material in nucleus. That's why he demonstrated. Now, in other words, we can say that cell stores hereditary material in the nucleus. That's how we can say what he did, Joachim Hammaring. So how he performed the experiment. He performed the experiment by using unicellular green algae of genus Acetabularia, which grow up to 6 cm as model organism of investigation. So individual algae consists of 3 parts: a distinct foot that contain nucleus, stalk, and the cap region. Now the algae is the largest unicellular organism, Acetabularia algae. This is the picture, and it can explain in short how we performed the experiment. We have 2 species, Acetabularia crenulata and Acetabularia mediterraneae. So, as you can see, the appearance of crenulata is different from the appearance of mediterraneae. Now, from here, he grafted different parts. That's why it is called the Hammaring grafting experiment. So he grafted this part, and from this part he grafted this part, transferring it to the foot region of Mediterranean species. So he grafted this part also. So as you can see, by grafting different parts, for example, this cap was removed and then was repressed. So this replacement of the cap, it is because the nucleus at the bottom controlled the development of this cap. So nucleus at the bottom controlled the development of this cap, and nucleus at the bottom here control the development of this cap. So nucleus in the base determines type of nucleus, type of the cap which is regenerated, because here the cap was removed. So the type of the cap which is regenerated depends on the nucleus in the bottom. So, or in the foot region. That's why then concluded that the nucleus contain the genetic material. For example, you can see this diagram. The cape is removed, then the foot region of acetabular crenulata is taken to this one, and then from there, this foot region control the development of the cape. And as you can see, this cape was removed. So after removing this cape, the nucleus at the bottom region controlled the development of the cape, and then the cape which was formed there, it was similar to acetabular crenulata. It is because the foot was joined with this stalk region. So you can see the cape was not developed according to acetabular mediterraneae, because this stalk region does not contain nucleus. So again, we have some of the diagram here. This developed intermediate type is because the nucleus here, grifted parts contain the nucleus of both acetabular mediterraneae and acetabular crenulata. But as you can see, how the grifts they were performed and the end products, what are the end product of this grifting. Again, as you can see, acetabular mediterraneae, you grift some of the stock of this acetabular crenulata, and then you put it at the acetabular mediterraneae. Then the final cup is similar to acetabular mediterraneae. Again, as you can see, the grifting of the acetabular crenulata, here you grift this upper part and then you dump it, then you put a stock from the acetabular mediterraneae. Then at the end of the day, you get the cup which is similar to acetabular crenulata. So since the nucleus was present in the bottom region and the nucleus determined the type of the cup which is formed, then we are concluding that the genetic material which control the growth of acetabular species, they are located in the nucleus. This marks the end of our session, and later I wish you nice studies.